So we have two sessions this afternoon. The first one's going to be on energy. The second one's going to be on emerging opportunities. The first session of the afternoon is going to be chaired by Gabe Palmer, our VP of Sector Development, and the second session on emerging opportunities by Rachel Ritchie. And we'll have a short break between those two. So again, for those of you that weren't here this morning, we're going to hold the questions till the panel discussion. And with that, let me introduce Gabe Palmer, that's going to introduce the this next session on energy.
So, before we get started here, in 2007, I think it was when Genome basically first started looking outside of academia, started looking at sectors, and we obviously looked at the health sector, we looked at the forest sector, fish and aquaculture, environment, and then Alan came in my office one day and said, what are we doing in mining and energy? I'm pretty sure they flippant came out of my mouth like rocks don't have genes or something like that. And the fact that we are here today and we're having a presentation on energy, and we could have done one on mining also, speaks volumes to how far the technology has come in its ability to be applied anywhere where living systems and biology is used. A whole bunch of reasons for that. A lot of it is traditional pressures. The cost of doing the work, the cost of doing the technology has come radically down, and it's, in many cases has gone out of the academic milieu as being applied everywhere there's biological systems. So I think we're going to hear three very different perspectives today. We're going to hear about a project from the perspective of somebody who's doing some very interesting systems types approaches to understanding living systems and how they apply to both energy and the mining sector. We're going to hear about somebody who's going to give a very more of a focused example of how genomics type technologies can be applied to better inform around spillage. And then we're going to have Ken from the BC Oil and Gas Commission talk to us just generally about the sector and about how innovation happens in that sector. So with that, I'd like to call up Steve there in the back. Steve is going to talk to us about a project that has been funded through the UP program, actually, from microbiomes to biorefineries and everything kind of that goes around that.
Okay, well, thank you very much for the invitation to talk about this work. Just a point of clarification. While this session is actually going to deal with some of those funded projects, what I thought I would just tell you a story that's a little bit more targeted to a recent Genome Canada funded project that Genome BC was a partner in that's winding down now, that led to some really compelling findings that I think you hopefully you'd be excited about, but actually changed the way I think about microbial communities in the natural world, not simply as agents of biochemical transformations in natural environments, but also as a paradigm for thinking about how we should design biological systems to transform matter and energy for industrial applications. So we're going to talk about from microbiomes to biorefineries in that context. And in the background here, it's hard to see, but I have a picture of a refinery. Okay? This is using fossil fuel to make all the things that we depend on. But there's a lot of interest in using biomass as a source of energy and as a source of materials, as a replacement for fossil fuels, a renewable, sustainable, more environmentally conscious way to drive modern society forward. And that is the conversion of lignocellulosic materials, specifically converting the tree parts, let's say, into sugar that can be used to produce fuels, but also monoaromatic or aromatic compounds that use precursors in the production of a variety of important materials, from plastics to pesticides, fertilizers, pharmaceuticals, that sort of thing. And there's a real compelling reason for this, in the sense that there's mandates all over the world to produce, in this case, more biofuel. And not just using food materials like corn, but also cellulosics that can be converted and replace petroleum that's currently driving our automobiles, for instance. And you can see that by 2022, we're looking at 16 billion gallons of produced biofuel, cellulosic biofuel. And that's leading to a huge capital investment in the production of biorefineries to convert that biomass into the fuel. And so billions of dollars all over the world are going to constructing these large plants that will operate like that refinery that I showed you at the outset, but in the production of more sustainable materials and energy resources. The problem, however, is that the cost of generating those cellulosic biofuels is not competitive at the present time. So this graph is basically showing you what it costs to make a gallon of biofuel from corn, ethanol, versus what it costs currently to do it from other cellulosic material. It's not cost competitive. And you can see a large fraction of the cost comes from enzyme. And so if we can bring the price point down on the enzyme side of things, we will make this production of cellulosics more economically viable and really transformative. The real issue in part is lignin, and getting the lignin out of the mixture, because the lignin can interfere with the conversion of the cellulose. The lignin itself is the source of those aromatics. So is there a way that we can transform lignin both to increase our yield of biofuel and bring the enzyme costs down, but to also create a new stream for materials? And so we turn to the microbial world to find metabolic pathways that can do this more efficiently. And so, I like to say that the microbial world possesses an infinite capacity to transform matter and energy. The genetic circuits evolved over 3.5 billion years are all around us. We just have to have a search function, some way of interrogating those microbial communities to find the answers. So we need a Google of sorts. You type in the molecule you want to make, and somehow, through some algorithm, you find the microbes or the genes necessary to produce the product. And one way that we started to do that is we can take the genomic information contained in any microbial community, whether it's from the human gut, from the ocean, from a forest soil. We can create large insert genomic libraries that contain anywhere from 30 to 60 genes. Often we get operons and gene cassettes from that. And we can clone that into a host like E. coli. And then we can conduct functional screens for the production. So you can think about this as the search function. The only thing missing, of course, is a sensor that's going to tell you if the product that you're interested in is actually being produced by the environmental DNA in E. coli. And so through this Harnessing Microbial Diversity for Sustainable Use of Forest Biomass Resources project, Genome Academy-funded project, we set out to conduct functional screens for lignin transformation using those types of environmental genomic libraries. We tried compound 7, which is a model lignin substrate that produced a fluorescent product when cleaved, and we couldn't get that to work. We tried DMP, which produces blue color. It's really good at detecting lactases and oxidases. We couldn't get that to work. And we tried using a fluorescence quenching approach on lignin, whole lignin. As it's being transformed, you get a decrease in the fluorescence profile, and we couldn't get that to work. And so we started to think, what can we do? So the idea came to us that we might be able to actually find a biosensor. So a fluorescent protein that's expressed in the presence of the degradation products for lignin, the aromatics themselves. And we could put that in co-culture with the environmental DNA. In the presence of lignin, the lignin is transformed, the biosensor will light up, and we'll find the clones that have that transforming potential. And the way that works is just outlined here. And what you can do with that is you can actually look for the transformation of lignin into monoaromatics, but you can also look for the production of monoaromatics. So you get the best of both worlds. You can look for biosynthesis, but you can also look for degradation. And we took an approach where we used an E. coli library, where the promoters of E. coli were fused upstream of green fluorescent protein. And we took 3 lignin aromatics, breakdown products of lignin, and we treated that promoter library with those compounds. And we found one promoter that responded. This is the PEMRR operon. That's driven by the PEMRR promoter. And then we used that to actually test in a variety of ways. It's a little complicated. In the interest of time, I'm not going to go into too much detail here. But the point is that we were able to show see that we could actually get a response. We use an enzyme characterized in the Altus lab that's able to break down lignin, and in the presence of that enzyme, we could get the biosensor to light up. But we couldn't get it to work with forest soils. We tried and we tried, we couldn't find the enzymes in the soils. So we turned our attention to coal beds. It's another Genome Canada-funded project where we had environmental libraries. We thought, well, coal might be a great place to look because it's just really old lignin. And there are not a lot of fungus living in the coal beds, but there are a lot of bacteria. So when we turned our attention from the screening point of view to the coal beds, we actually found a number of activities that could transform the lignin. In fact, we found 24 clones of a relatively small set of libraries that were able to convert lignin polyaromatic into monoaromatics that lit up the biosensor. And this is just showing you on different forms of lignin, softwood lignin as well as high-performance lignin, that we get different monoaromatic profiles for a variety of the clones. And so things that are blue are not active. The bluest is no activity, no transformation into that particular monoaromatic, and things that are pink or purple show that we're getting that particular monoaromatic in the GCMS profile. So we're excited because we can see different types of profiles based on different clones that we had identified from the coal beds. We could go on to show that we could produce things like vanillin very nicely using a standard. We could also clone out individual enzymes. To validate the screen, we cloned out a lactase, COP8 homolog, and showed, you can see in the presence of copper, that we could get the lignin transformation profile in blue. You can see over there. So we actually were able to show that this approach is viable. So we've got a search function. So this just shows you all the clones that are characterized and the relationships connecting. It's a surface plot. The connections show homology. We've outlined in color the genes that we think are involved in the lignin transformation process. And recurring, there were 6 functional classes that occurred on these different environmental clones that we could implicate in the process. So that's very interesting. It takes a set of different enzymatic components working together in different ways to produce these lignin transformation profiles in the environment. When we looked more closely, many of them were associated with mobile genetic elements. So phage or plasmid genes were associated with these 6 functional classes on the active clones, which led us to think of a model in which lignin transformation is an adapted trait in the coal bed, and that these cassettes are rearranged in different ways to produce different monoaromatic profiles that produce carbon resources for the microbial communities. And so they're interactive, and they're mobile, and they synergize in different ways. And that led us to think more about a model for microbial community metabolism in which cooperation is integral. And so you can think about a metabolic pathway, ABCD, in one cell. What we're suggesting is actually distributed among multiple cells. And we'd like to bring that concept into the biorefinery, either by creating gene cassettes in which we recombine those genetic elements to produce specific monoaromatic profiles for the biorefinery, or, in fact, we create pools of clones that each produces a different outcome that synergize in combination. This has led us to start a company called MetaMixes, in which we're actively pursuing these outcomes. And if you want to learn more about the power of microbial cooperation, I point you in the direction of a recent TEDx talk that I gave in October. So with that, I'd like to end, and I'd also like to thank the collaborators at the University of British Columbia, these 2 of which are in the audience, Bill Moen and Steve Withers, who participated in this project, and also highlight Lindsay Eltis, whose lab did a lot of the biochemical purification. So on that note, I'll end and answer any questions during the panel, I think.